So muscle or gland, motor neurons, which is bringing the motor command from the motor cortex of the cerebral hemisphere in the subcortical parts. It may be brainstem, it may be spinal cord. When it is in spinal cord, it is known as corticospinal fiber, which I will talk later, and corticobulbar or corticonuclear fiber in case of brainstem, and it is having motor commands for the glands and the muscles. These neurons bringing the impulse for motor command from the cortex to the subcortical areas and spinal cord are known as upper motor neurons. Upper motor neurons, and there are motor neurons in the brainstem in form of cranial nerve nuclei, and there are nuclei for motor neurons in the ventral or anterior horn of the spinal cord. These neurons are known as lower motor neurons. So two types of motor neurons: upper motor and lower motor neurons. So in case of sensory neurons, first order or first degree, second degree and third degree. First order, second order, third order neurons. And when I will talk about the motor neurons, these are upper motor neurons and lower motor neurons. The function of CNS is collecting sensation, integrating, interpretation, processing of the senses, and forming motor command. So interneurons are seen in central nervous system. These interneurons collect information from sensory neurons, integrate the information, compare with the previous memory, interpret the information, and it process the information and form a motor command. And motor command is carried by the motor neurons to the effector organs. So what is nervous system? Nervous system is a stimuli response system. Stimuli is received by and response is made by these motor neurons, and integration, interpretation and processing is done by these interneurons in the central nervous system. Interneurons are neither sensory nor motor. These are independent neurons and form many thousand connections with the other neurons in the CNS. I have already told neurons are contiguous, neurons are not continuous. As I have already told, students which are very new medical students may be overwhelmed by intricacy of the nervous system, but be patient. Treat this neuroanatomy or neuroscience as a foreign language. Before be fluent in the spoken foreign language, please be master in the vocabulary and grammar. So before understanding the neurosciences and neuroanatomy, I have briefly or concisely talk about the development of the nervous system. In the third week of development, in the last part of third week of the development, there will be a well-formed trilaminar disc. It is having, as the name indicates, trilaminar. It is having trilamina or 3 layers: ectoderm, mesoderm and endoderm. It is having a primitive strip and primitive node, and here is buccopharyngeal membrane. Primitive strip, primitive node also known as Hansen node, and this is buccopharyngeal membrane. Between the primitive node and the buccopharyngeal membrane, there is a solid rod-like structure known as notochord. So it is primitive strip, primitive node. Here is buccopharyngeal membrane, and here is developing solid rod-like notochord. In the end of 3rd week and in the first hours or days of the 4th week, there will be modification or differentiation of the ectoderm known as surface ectoderm, and above the notochord, this surface ectoderm will modify to form the neuroectoderm. So here is neuroectoderm above the notochord. This neuroectoderm will form a plate-like structure like this, and this plate is known as neural plate. So above the notochord, here is notochord, and here is the developing neuroectoderm. It will form the developing neural fold or neural plate. Initially it is plate like this. This is notochord and this is neural plate. As time passes by and by growth of the mesoderm, underlying mesoderm, this plate will form a neural fold, and the lateral margins of this neural plate will elevate. I will section this from here. In section we can see here is the neural plate, and here is developing surface ectoderm, and here is notochord. As time passes by, and here is mesoderm, paraxial, intermediate and lateral plate mesoderm, again paraxial, intermediate and lateral plate mesoderm. So as time passes by, the growth of this mesoderm, these folds of this neural plate will elevate and form a neural groove. So it will form a neural groove like this. Notochord and mesoderm. And here is the surface ectoderm. Now this part which was neural plate, now it will fall a neural groove here and neural fold here. So these are elevated parts of the plate are known as neural folds, and this part is known as neural groove. As time passes, like part meet the like part, the surface ectoderm will meet the surface ectoderm, neural folds cells will meet the neural fold cells, and the margins of neural groove or lowermost part of the neural fold will meet to form a tube. This tube, now known as neural tube, and this tube will develop the CNS that is brain and spinal cord, and some part of the peripheral nervous system. And here is again our paraxial, intermediate and lateral plate mesoderm. Now, this paraxial mesoderm will start segmentation. These segments are known as somites. At the fourth somite stage or 20th day of the development, the folds of this neural groove will start migrating dorsomedially to form a neural tube. It will start from the middle part of the neural groove and it will spread rostrally and caudally. On 20th day of development, that is 4th somite stage or 20th day of development, and this fusion will be completed on 23rd or 24th day, and this tubular structure will form neural tube. This neural tube is having anterior opening and posterior opening. This anterior opening known as anterior neuropore and this posterior opening known as posterior neuropore. This anterior neuropore is closed by mid of 4th week, that is 25th day, or fused by 20 somite stage, and this posterior neuropore fuses by or closed by 28th day or on the 25th somite stage. When the anterior or posterior neuropore are not fused, these will lead to a very serious form of neuronal development defect known as anencephaly or gastrochiasis. So it is anencephaly or the other types of neural tube malformation. Now I will show you the section of this neural tube. Initially this neural tube is having a single layer which are columnar. Initially this neural tube will show a single layer of columnar cells resting on the basement membrane, and it is having an outer and inner limiting membranes. These cells are known as ependymal cells. These ependymal cells will form the pseudo-stratified cells. As time passes by, these cells are converted in pseudo-stratified cells, and as time passes by, these cells will form stratified cells. These cells which were columnar will form pseudo-stratified cells, and as time passes by, these cells of ependymal zone will multiply and migrate outward to form two types of cells. So cells migrating outside, divide and migrate, divide and migrate, divide and migrate. These cells are of two types. One is neuroblast and other is spongioblast. These neuroblasts will form the neurons or the conducting system of the nervous system, and this spongioblast will form the non-neuronal cells, non-nervous cells or glial cells, that is oligodendrocytes or oligodendroglia, astrocytes and the other types of connective tissue cells of the CNS. And this zone, now this is ependymal zone towards the cavity. This is cavity of the neural tube. This cavity will form the ventricular system of the brain and center canal of the spinal cord in later part of the development. And it is ependymal zone which will cover the central cavity and the ventricular system in the later part of the development, and the middle part is known as mental zone. This middle part is known as intermediate zone or mental zone, and outermost part, it is known as marginal zone, marginal, and ependymal zone is known as matrix or ependymal zone, matrix or ependymal. So developing neural tube is having three layers. Innermost is ependymal zone or matrix zone. Intermediate zone known as mental zone, which is having two types of cells. One is spongioblast, other is neuroblast. Spongioblast will form the glial cells or the sustentacular cells or supporting cells, and it is having neuroblasts which will form the nervous cell of the CNS. And outermost zone known as marginal zone, it will form the white matter of the central nervous system. What is white matter? What is gray matter? What is a neuron and what is myelination? All these topics I will cover in my next lecture. So it is having three zones. Innermost zone known as ependymal matrix zone, intermediate zone which is having spongioblast and neuroblast, and outermost zone which is form the white part of the brain and spinal cord known as marginal zone. So it is having a tube-like structure which will start two constrictions and dilatation in its rostral part during the 5th week of development. So anteriormost part which will dilate, there will be a constriction, again constriction, and again dilated part, and posteriormost part of almost similar diameter, elongated part. The posterior part of neural tube will develop in spinal cord. The anteriormost big dilatation or vesicle will form prosencephalon or forebrain. The middle vesicle will form midbrain or mesencephalon, and the posteriormost dilatation or the vesicle will form the hindbrain or rhombencephalon. So in end of 5th month, there will be 5 dilatations and more constrictions. I will draw a different diagram here. So here are more dilatations or vesicles. Long spinal cord. So prosencephalon, it will form 2 vesicles. One is telencephalon, a small part diencephalon. The middle part, it was mesencephalon. Most primitive part will remain as mesencephalon, and posteriormost part, which was rhombencephalon, will form 2 vesicles in form of metencephalon and caudalmost myelencephalon. So this is developmental part of the CNS. Now what happens? These primary vesicles will form 3 primary vesicles: prosencephalon, mesencephalon, rhombencephalon, will form 5 secondary vesicles: telencephalon, diencephalon, mesencephalon, metencephalon, and myelencephalon. And here is beautiful long cylindrical spinal cord. So when brain is fully developed or CNS is fully developed, it is having a structure like this. When we see the lateral section of the spinal cord and brain, it will be like this. So here is telencephalon, it will form the cerebrum. Diencephalon, it will form the diencephalon part having thalamus, hypothalamus, metathalamus, epithalamus, and subthalamus. 5 parts of the diencephalon. This mesencephalon will form the midbrain. So here is cerebrum formed by the telencephalon. Here is diencephalon, and it is having thalamus, hypothalamus, metathalamus, epithalamus, and subthalamus. Midbrain, mesencephalon will form midbrain. This metencephalon will form the cerebrum and the pons, and myelencephalon will form the medulla, and remaining lower part of almost same diameter cylindrical part is known as spinal cord. This is spinal cord part. So what we see, the 5 secondary vesicles will form the brain, and the posterior or caudal part of the developing neural tube will form the spinal cord, which retains its primitive structure in form of a tubular structure. So this is the development of the brain. Here I will discuss very briefly about the formation of the nuclei of the functional components of the nerves. in primitive part of neural tube, that is spinal cord. Here is ependymal zone. This is intermediate or marginal zone, and here is outermost mental zone. So mental zone is having the nuclei of the spinal nerves. It is having 4 components of 4 types of nuclei, and this part of the neural tube is known as sulcus limitans. So this is dorsal part, this is ventral part, this is part of developing neural tube is known as roof plate, and this part is known as floor plate. The roof plate is having alar lamina and the foot plate is having basal lamina. Alar lamina will form the sensory neurons and basal or ventral lamina will form the motor neurons. So motor neurons will develop in the basal lamina or towards the roof plate, floor plate, and alar lamina towards the roof plate will form the sensory nuclei. So here are, these all are general senses and general motor outflow in case of spinal cord, which I will talk in detail in my lecture on brainstem and spinal cord. Now after a break I will continue the introduction of neuroanatomy.